This diagram here might look pretty, might look complicated. This is protein synthesis, and that's the next thing we're going to talk about. In previous videos and in previous topics, we talked about DNA replication, we talked about cell replication, we talked about the generation of genetically individual gametes and how that can create a huge degree of genetic variation in offspring. Now we want to look at, okay, you've got that genetic variation there, but how is that expressed? How does that result in different looking people, different processes? How does this fancy, amazing molecule, DNA, that's inside our cells control so much? And that is all beginning with the process of protein synthesis. So that's what we're going to cover in this video. This diagram here is a nice, simple summary of the process, starting off in the nucleus and moving into the cytoplasm. It's made up of two main stages, the first being transcription, the second translation. We're going to talk about each of those. Before we do that though, some definitions for you. When we're looking at our DNA within the cells, we already know that a gene is a particular section of DNA within a chromosome that codes for a particular trait, particular thing. When we put all of those genes together, all of those genes amongst all those chromosomes, then the total of all of that DNA in all of the cells, in all of an organism's cells, nuclear, mitochondria, chloroplast, if you're a plant, that sum total of all that DNA is known as a genome. And that genome is the recipe book for that organism. There is a lot of effort around genome mapping these days, because if you can map an entire genome, then you could theoretically find out what every gene does and find out all of the parts that creates this particular organism. So we know what a gene is. Now we know what a genome is. It's the entirety of that DNA. You can see in that diagram there, we've got genes. Genes all together make up chromosomes, and then those chromosomes all together make up the genome. And of course, when we go lower than a gene, we've got DNA, which is made up of nucleotides. We already know this about genes, as I said. And one thing that I feel is really important to sort of set the scene here for the main focus, I suppose, of this upcoming chapter is that genes code for a particular trait, and they can be controlled. They can be switched on, they can be switched off. And this is what we call gene regulation. So when a gene undergoes the process of protein synthesis, that is effectively equating to it being read and understood, then that's known as gene expression. We can control that through gene regulation, both of which are the processes we're going to be looking at in this chapter. This should be familiar to you, the universal genetic code. You can see here we're using our mRNA, effectively our codons, our three base sequences will code each for an individual amino acid. You can see here that some amino acids have multiple codons that code for them. So when protein synthesis is happening, the recipe is written in the genetic code in the form of codons. We run along the genetic code looking at these three base sequences, the codons, and translating them into amino acids. As we do this, we're producing lots of amino acids which attach together, and when we've got multiple amino acids, we've got a protein. So protein synthesis is the process of basically writing down that genetic code, transcribing it, that's transcription, our first step, taking it away and then translating it. I always use the analogy of copying a recipe off your grandma. Let's say your grandma makes an awesome spag bowl. You might go over to her house and ask her for a recipe. She would copy that down for you, and that copying down process is known as transcribing. And then you take that recipe away, you take it to your own kitchen, okay, and you read it through and you follow the recipe. And at the end of it, you're going to have an awesome bag of spag bowl and a bowl of spag bowl. You can eat it out of a bag if you want. And that process is known as translation. You're translating that recipe into the food that you want it to be. And that is the way I like to think about protein synthesis. So the way that DNA controls all of life and is the recipe book for us is by being read and translated into proteins. It's the way that genes are expressed. When we look at our DNA, it is effectively a template or master copy, you may say. We don't actually use the DNA itself to be read and make the amino acids. So the DNA, much like in DNA replication, it will split open and will make a new chain of nucleic acid there using the DNA as a template. But instead of it being new DNA like it is in DNA replication, in this case, we're making RNA, which you'll remember is single-stranded, uses uracil instead of thymine, and it uses ribose sugar instead of deoxyribose sugar. And that particular type of RNA we're looking at is messenger RNA. That messenger RNA, which is copied off the DNA as a template, is then the instructions that we're going to use to make our proteins. That moves from the nucleus into the cytoplasm. And all of this is done by complementary base pairing, which is why we ram it into you in class, ATGC or GCAU. Okay, remember we live at the Gold Coast or Gold Coast, Australia, AU. Because without understanding complementary base pairing, you don't understand DNA replication and you won't understand protein synthesis. Because the DNA, as simple and complex as it is simultaneously, is the copying process is very straightforward. It sees an A, it puts a U. It sees a T, puts an A. It sees a G, puts a C, etc. So transcription is this first process. To summarize, transcription is making this mRNA, which is our copied instructions. So transcription is a process of copying down the genetic code by creating a strand of mRNA. And that strand of mRNA we call the transcription unit. You can see in this simplified picture, basically, we've got some parent DNA here. Well, I suppose it's not parent DNA in protein synthesis. We've got a DNA strand. We want to copy that gene. Okay, let's say that we've got a cell that wants to make some insulin. So we want to copy the gene for insulin. The chromosome unwinds, the DNA opens up, and some mRNA is made that is complementary to the gene that we want to copy. That transcription unit, that copy of the gene, will then leave the nucleus where it can go on and go through other processes. So the gene that contains a transcription unit, as I said, let's use the example of insulin, will unzip, untwist, and it'll open up. RNA polymerase, very similar DNA polymerase, but working with RNA or to create RNA, will add free-floating nucleotides to the DNA strand, creating mRNA. Same as we had with DNA replication, but this time we're making RNA. Okay, and you can see that process happening there. So very, very similar to DNA replication. It's unzipping, unwinding, it's opening up, and RNA polymerase is creating a strand of mRNA using complementary base pairing, which is what you can see here. Where there's a C, we put a G, and so on. I just noticed there is an error in that copying there. There's a little cheeky mutation for you. We'll learn about that later on. As that RNA polymerase moves along and generates the mRNA, it also rezips the DNA so that it gets rewound up, or the DNA rezips itself, and the DNA protects itself again, because we know that DNA is vulnerable and exposed when it's unbound. So the mRNA is being produced by the RNA polymerase as it's open, and then it seals back up once that's copied. Once the mRNA copy of the transcription unit has been created and is finished, it then immediately detaches so that DNA can wind back up and go back into its standard state. Now, what we produce from this, the mRNA, is unfinished. Okay, it's not ready for plating. We need to fix it up a little bit. It's what we call pre-mRNA when it's directly copied. So the pre-mRNA it's got all the right bits, but we need to tidy it up. It's made up of DNA known as both introns and exons. Exons are what we know as coding DNA, which are the sections that contain the information we're going to need to make the protein. Introns are non-coding DNA. Used to be called junk DNA, is no longer because we realize it's not junk. And the non-coding DNA, the introns, through a process of splicing, are removed. And the exons join back together. What we have at the end of that is what we call mature mRNA, which can then move on to be translated. So you can see this here. We've got those red or pink sections, which are our exons, and then the uncolored sections are the introns. As we move from pre-mRNA to mature mRNA, the introns are cut out, and then the exons are stitched together, and that process is known as splicing. This process is not as simple as it may look, and the introns aren't just completely wasted. They actually will direct the way that the exons are put back together. So the first section you see won't necessarily always be first. It might get moved around depending on the need. Before it can leave, because we need our mature mRNA to leave the nucleus into the cytoplasm for any sort of translation to occur, then it needs it needs a hat and boots. So it puts a guanine on its head as a protective cap. Okay, so it puts an extra G on the end, and then it has a poly-A tail, which can be made up of hundreds of adenines. So that poly-A or polyadenylated tail helps protect it as well. Puts a G on top, a guanine cap, and then a poly-A tail. As I was just saying, the introns that are the non-coding DNA direct that splicing process. So the exons from the same pre-mRNA can be sliced differently and giving us different versions of that transcription unit and different versions of the resulting protein. So the introns used to be called junk DNA because they thought they just got cut out and thrown away and they didn't do anything. They were just filler. It's not the case. They're actually the boss here directing how that process is occurring. Okay, so at the end, we might only have exons, but the introns are the ones that control that end product. We're almost at our next stage, translation. So we have, at this point, taken DNA and, to summarize, made a copy in the form of mRNA. The mRNA, messenger RNA, now needs to leave the nucleus into the cytoplasm, where it will meet up with our ribosomes, which are protein builders. We learned about ribosomes way back at the very start of year 11 when we learned about cells and their basic structures. So ribosomes are protein builders, protein synthesizers. They are the site for construction of our proteins. So that's where our next stage, which is translation, occurs. At the ribosome, the mRNA, which is fed through the ribosome, the analogy here is like letters into a letterbox, probably, I don't know, more like moving something through a pipe, I suppose. So the mRNA is fed through the ribosome, and inside the ribosome, we have binding sites where mRNA can meet up with a fellow RNA, tRNA, and that's where translation occurs. So we've got two parts to the ribosome. We've got the 40S subunit, which decodes the mRNA, and then the 60S subunit, which creates peptide bonds to join the amino acids together. So the 40S subunit is effectively where the translation is occurring. When we are looking, and hopefully you remember this from previously, when we're looking at our mRNA, we're looking for a particular codon, for where it will begin. So protein synthesis, in terms of translation, will not kick off until the ribosome sees AUG. AUG is the start codon, and once it sees that, that is the signal that it's time to start making a protein. So that mRNA is feeding through the ribosome, and as soon as we hit an AUG, which every gene should start with an AUG codon, then it starts translating. So once in there at that 40S subunit, as we said, the mRNA can interact with the tRNA. tRNA has a super funky little structure here. Now it is single-stranded, even though it's folding back on itself, so it is still an RNA molecule. And it has two really important parts to it that are crucial for protein synthesis. Okay, it has an anti-codon. Anti, we always know, means opposite, and that's a 3-base sequence which is complementary to the base pair of the codon. Okay, so if we are reading a codon on our mRNA that is GCC, then the anticodon on the tRNA must have CGG. The reason that we must have an anticodon is because it needs to temporarily form a complementary base pair. And in order to do that, it needs to be complementary. So the anticodon needs to be the opposite, okay? Much like you can't plug your phone charger into another phone charger, okay? You need to plug it into the opposite of it, which is the charging port. Now, the codon at the bottom will code for a particular amino acid. The anticodon will be opposite to that. And then at the top of our tRNA, we have an amino acid, which is attached via an ester bond. You don't need to know what that is, although, you know, a spoiler alert for the chem kids, you'll be learning about that soon. That amino acid is held on at the top there, and that's the amino acid that is coded for by the codon on the mRNA. Once the mRNA and the tRNA find each other, they find a match, then what happens? So we have the anticodon, the complementary base of our codon, and they will match up together and they will temporarily form a bond where they will link up. Once that happens, the tRNA, recognizing that bond, will release the amino acid. Okay, so if the amino acid it's holding on to is methionine, the tRNA is holding on to MET, methionine. The anticodon will read UAC because that's a match to AUG, which is what methionine actually codes for. Okay, so the anticodon UAC there will match with AUG, which you see codes for is the codon for methionine, and that is the amino acid the tRNA will be holding on to. As that mRNA is fed through, the tRNA with matching codons will come and it will bind with it. Okay, once it's found its match, the tRNA will basically immediately detach once it's found its match, and then it will move on, leaving that amino acid behind. That amino acid will then bind to the next amino acid that is released by the next codon and tRNA, and then the next and the next. And as these amino acids are connecting to each other, we have an amino acid chain known as a polypeptide chain. This is the basic form of a protein. These tRNA will be recycled. They'll go off into the cytoplasm of the cell, and they will later on pick up the amino acid that they're coded for, and they can recycle that for later use. This whole process will continue until our entire mRNA molecule has been decoded or translated, and what we have left is a very long polypeptide chain. I think the simplest, shortest protein ever discovered in the scientific communities are about 50 amino acids long. Most are 250 amino acids plus. That tRNA, as I said, gets recycled and used again, and that polypeptide chain will then be taken to our endoplasmic reticulum, which is another organelle of our cell you should know, where it can then be processed further and folded to make a protein. And that's the last step. So just to summarize, transcription is happening in the nucleus thanks to RNA polymerase creating an mRNA copy of the template DNA, and that copy is known as our transcription unit. It's a copy of the gene we want to express. So if you've got a cell that wants to make insulin, then it will copy or transcribe from the gene for insulin. The mRNA gets processed going through going from pre-mRNA to mature mRNA before it leaves the nucleus. It then leaves the nucleus into the cytoplasm where it attaches to ribosome and moves through the ribosome where it meets up with its BFFs, the tRNAs, where the codon on the mRNA will bind to the anticodon on the tRNA. As that happens, it releases amino acids. Amino acids form peptide bonds between them, forming a long chain we know as a polypeptide. After this, the polypeptide needs to go through some processing to finalize itself as a protein. So protein synthesis is producing that primary structure of a protein, the amino acid polypeptide chain. After this, we have 3 levels of structural change. Not all proteins will go through these steps. It just depends on how complex the protein is. So secondarily, we will have pleating, or it will spin or twist to make an alpha helix. So we have an alpha helix or a beta pleated sheet, depending on whether it gets that pleating corrugated look or whether it twists to form a helix. And then we can get the tertiary structure where we have other interlinking between amino acids in the tertiary protein. So the secondary protein structure, this is all sort of extra information, by the way. The secondary protein structure is as a result of hydrogen bonds between the amino acids, and the tertiary protein structure is a result of other types of complex bonding occurring between the side chains of the amino acids. Then lastly, if you want a really complex protein, we can twist it around another protein to make the quaternary protein structure. Okay, hopefully that made sense to you. If not, there are some great infographics out there, and also we'll be going through it in class. Hopefully you made some great notes, and yeah, we'll see you for the next video all about gene expression.